Hello, my name is Kaylie Wheeless, and I am a student at Henderson State University. Today, I am going to be presenting our project titled The Analysis of Microbial Organisms and Their Interactions Within a Unique Cave System in Central Tennessee. I'd like to thank my teammates and mentors for their help with the project. These three environments are typically used as potential models for life on early Earth. The first is a deep-sea hydrothermal vent, far beneath the surface of the ocean, where sunlight is not available and therefore photosynthesis does not occur. The second is a hot spring, where the water is superheated to the point that oxygen is actually insoluble, creating an anoxygenic environment. And the third are caves, where photosynthesis and its products may be available at the opening, but not far beneath the surface. Despite this, each ecosystem supports its own biologically diverse organisms. Our team has been studying a cave located in the Highland Rim geologic region of Central Tennessee, called Secret Squirrel Cave. It was first entered in 2005 and has currently been mapped to a distance of over 15 miles. The cave contains a stream, which runs into an overflow pond located in a passage of interest called the Petroleum Passage. The Petroleum Passage was named because of its strong petroleum smell. The overflow pond in the passage has a sandy bottom with dark patches. These dark patches occasionally produce an oily substance that rises to the surface and bursts, known as tar balls, and these are responsible for the petroleum smell within the passage. This pond is also ringed by bands of colored sand, indicative of microbial activity. Initial sampling was conducted on May 26, 2019. At this time, 30 samples were collected from areas of microbial interest, including active speleothems, the water, oil droplets, tar balls, colored rings of sand, and floating calcite rafts within the passage. These were collected using sterile cotton swabs, which were then placed in sterile centrifuge tubes broken off and capped to avoid contamination. These were then iced for transport and stored at negative 80 degrees Celsius until processing. Samples were prepared for metagenomic sequencing by purifying and extracting the DNA present. The concentration of DNA was then estimated using a nanodrop 2000 spectrophotometer. Metagenomic sequencing was done commercially by Molecular Research LP located in Shallow-Water, Texas. There, the 16S rRNA sequences from each organism present in every sample were isolated and compared to the 16S rRNA sequences of known organisms present in the BLAST database. The percentage match then determined what operational taxonomic unit they were placed in. A match of 97% or more indicated a species match, 95 to 97% indicated genus, 90 to 95% indicated family, and so on. Today, 9 of the 30 initial samples have been analyzed, revealing some Archaea, but also unique taxa including methanotrophs, sulfur and ammonia oxidizers, eubacteria, Archaea, thermophiles, and organisms that degrade hydrocarbons, as well as other unique taxa. And then here are the results of our metagenomic sequencing. You can see that 59 operational taxonomic units matched at the species level, 236 at the genus, 123 at the family, and 76 at the order or higher. And then from here, these are the samples that we processed. You can see that the rings of sand actually host the most unique operational taxonomic units, as well as the most organisms. And then here are a few that were especially interesting. There were many ammonia oxidizers, as well as thermophilic bacteria and Archaea. And occasionally there were some organisms that were found that are commonly found in deep-sea hydrothermal vents. In the future, we would like to conduct a more targeted sampling and analysis, and then we would also like to conduct tests on the gypsum formations. We would like to conduct salamander identification. We believe these salamanders may actually be feeding on the bands of colored sand containing organisms that can process the tar present in the ponds. Our team is currently waiting on the standard water chemistry test and the hydrocarbon analysis.